Today we are going to discuss about carbohydrates. Carbohydrates are basically organic molecules, one of the various macronutrients which is required for sustaining our life. Carbohydrates contain basically carbon, hydrogen, and oxygen, and they are found in a wide range of foods that bring a variety of other important nutrients to the diet. For example, vitamins and minerals, phytochemicals, antioxidants, and dietary fiber. Though dietary fiber is not providing any nutrient, but it is very important part of our diet which must be consumed daily, because first thing is it provides bulk to the diet, and second thing it also plays important role in prevention of many diseases, specifically hypercholesterolemia leading to ischemic heart disease. The basic molecular formula of carbohydrate is CH2ON, where N may be 3 or more than 3. When sugar is heated at high temperature, final residue is carbon along with water. Means these are final result is hydrate of carbon, which may be one of the region of its name as carbohydrate. By definition, carbohydrates are simply polyhydroxyaldehydes or ketones, that is, containing functional group either as aldehyde or as ketones. And these are classified in three broad categories, that is, monosaccharides, oligosaccharides, and polysaccharides. Monosaccharides, as name is self-explanatory, contains single polyhydroxyaldehyde or ketone units which cannot be hydrolyzed further as it is smallest monomer unit. For example, glucose, fructose, galactose, etc. Oligosaccharides contain 2 to 10 monosaccharides units which are linked together by glycosidic linkages. For example, sucrose, maltose, lactose, stachyose, raffinose, etc. Polysaccharides are made up of more than 10 monosaccharide units. For example, starch, glycogen, cellulose, etc. Now let us see major functions of carbohydrates. They form major organic matter on the earth because they have to play extensive roles in all forms of life. They also serve as energy stores, specifically in the form of starch in plants, whereas in the form of glycogen in animals, specifically in liver and skeletal muscles. They also play important role to supply fuels in the form of energy for different works to be done by the body, and metabolic intermediates. Ribose and deoxyribose sugars are important component of RNA as well as DNA. They also provide structural elements for the cell, also bacteria as well as plants. They are linked to many proteins and lipids to play key roles in mediating interactions among cells and interactions between cells and other elements in the cellular environment. Now let us see structure of important carbohydrates. First of all, we will discuss about monosaccharides. Monosaccharides, as you know, may contain 3 or more carbon units, and depending on number of carbon units, it is named. For example, if it contains 3, 4, 5, 6, and 7 carbon atoms in their backbones, they will be named like triose, tetrose, pentose, hexose, and heptoses respectively. Their name also depends upon different functional groups. If it contains Aldehyde group, it will be named as aldotriose, aldotetrose, aldopentoses, aldohexoses, and aldoheptoses. If it contains ketone groups, it will be named like ketotriose, ketotetroses, ketopentoses, ketohexoses, and ketoheptoses, respectively, for containing 3, 4, 5, 6, and 7 carbon atoms in their backbones, respectively. Carbons of sugar are numbered beginning at the end of the chain nearest to the carbonyl group. All the monosaccharides except dihydroxyacetone contain one or more asymmetric carbon, that is chiral carbon atoms, and thus occur in optically active isomeric forms. Here you can see basic structure of smallest monosaccharides, that is aldotriose. Example is glyceraldehyde. Both isomers are given here as D-glyceraldehyde. Here hydroxyl group is on right side, whereas in case of levorotatory glyceraldehyde, hydroxyl group is on left side. This is structure of ketotriose, that is dihydroxyacetone having ketone group as functional group, whereas glyceraldehyde has aldehyde group as functional group. Here you can see representative monosaccharide unit structure. Here D-glucose and D-fructose are examples of hexoses. D-glucose is having 6 carbon unit in their backbone having aldehyde group as functional group, whereas D-fructose has also, being hexose, has also 6 carbon atoms in their backbone, but it has ketone functional group. There is also example of 2-deoxy-D-ribose. Ribose is basically pentose. It is deoxygenated at 2 carbon unit. You must know that in aqueous solution, all monosaccharide units occur as cyclic or ring structure in which carbonyl group forms a covalent bond with oxygen of a hydroxyl group along the chain. The formation of these ring structures is the result of a general reaction between alcohols and aldehydes or ketones functional group to form hemiacetals or hemiketals, which contain an additional asymmetric carbon atom and thus can exist in 2 stereoisomeric forms, that is alpha and beta, which we will discuss diagrammatically also in next slides. For example, D-glucose exists in solution as an intramolecular hemiacetal in which the free hydroxyl group at C5 has reacted with the aldehydic C1, rendering the later carbon asymmetric and producing 2 stereoisomers like alpha and beta isomer. Isomeric forms of monosaccharides are known as anomers, which differ only in configuration about hemiacetal or hemiketal carbon atom. Hemiacetal or carbonyl carbon atom are called anomeric carbon. The alpha and beta anomers of D-glucose interconvert in aqueous solution by a process called mutarotation. Here you can see the cyclic structure of glucose as well as fructose. As we have already discussed that in case of glucose, as for example, you can see this free hydroxyl group at C5 will react with aldehyde C1 and rendering the later carbon asymmetric, that is C1 will behave as asymmetric and producing 2 stereoisomers as alpha and beta. Here you can see that there is carbon number 1, which is responsible for its isomeric form as when this hydroxyl group is on lower side is known as alpha D glucopyranose, and when it is on upper side it is known as beta D glucopyranose. Similarly, in case of deep fructose, it forms 5-membered ring structure, that is furanose. And here also C2 is responsible for its isomeric forms, that is alpha furanose as well as beta furanose. Monosaccharides can be oxidized by mild oxidizing agents like ferric or cupric ions. Carbonyl carbon is oxidized to carboxyl group. Glucose and other sugars capable of reducing ferric or cupric ion is known as reducing sugar because it will reduce this ferric ion to ferrous ion and cupric ion to cuprous ion. This property is basis of Fehling's reaction, which is a qualitative test for measuring presence of reducing sugar in the sample. Here, amount of oxidizing agent reduced by sugar solution will tell the amount of concentration of sugar present in that medium. Two sugars that differ in configuration around one carbon atom is known as epimers. In next slide, we will see that D-mannose and D-glucose are epimers because there is difference in configuration around carbon 2, whereas D-galactose and D-glucose are having a difference in configuration around carbon 4. That's why they are also epimers. But D-galactose and D-mannose are not epimers themselves. Here you can see the basic difference in configuration at C2 of mannose and D-glucose. So they are epimers. And there is difference in configuration of C4. This is hydroxyl group is on left side, and in case of glucose, it is on right side. So D-glucose and D-galactose are epimers with each other. But these D-mannose and D-galactose have no relation of epimer like this. Both of them are epimers of D-glucose only. Now let us see structure of disaccharides. In case of disaccharides, there are two monosaccharide units which join covalently by an O-glycosidic bond and form disaccharides, where a hydroxyl group of one sugar reacts with the anomeric carbon of the other. Here you can see anomeric carbon of galactose combines with C4 hydroxyl group at C4 of glucose, forming beta 1,4 glycosidic linkage in case of lactose, whereas in case of sucrose, there is anomeric carbon of glucose linked with C2, hydroxyl group of C2 of fructose, forming alpha 1,2 glycosidic linkage in case of sucrose, which contains one molecule of glucose and one molecule of fructose. In case of maltose, there is alpha 1,4 glycosidic linkage, which is prepared by combination of two molecules of glucose. In case of cellobiose, there is linkage of beta 1,4 glycosidic linkage between two molecules of glucose. So these are important disaccharides. The oxidation of sugar's anomeric carbon by cupric or ferric ion occurs only with the linear form, which exists in equilibrium with the cyclic forms and not in the cyclic forms. When anomeric carbon is involved in a glycosidic bond, that sugar residue cannot take the linear form and becomes a non-reducing sugar. As we have already discussed that reducing property exists only in the linear form of the sugar. In disaccharides or polysaccharides, the end of a chain with a free anomeric carbon is known as reducing end. Now let us see structure of polysaccharides. Polysaccharides are basically of two types, that is homopolysaccharides having single type of monomers throughout the chain, whereas second is heteropolysaccharides which may contain two or more different kinds of monomer units. Some homopolysaccharides serve as storage forms of monosaccharides and used as fuels to provide energy. For example, starch is storage form of polysaccharide present in plants, whereas glycogen is storage form of storage forms of monosaccharides. An important polysaccharide present in animals, specifically in liver and skeletal muscle tissues, to provide energy in the time of emergency. For example, in case of fasting, when blood glucose level is reduced to provide much of energy required by the body. Other homopolysaccharides serve as structural elements. For example, cellulose, which is structural element of plant cell walls, and chitin, which is structural element of animal exoskeletons. Heteropolysaccharides provide extracellular support for organisms of all kingdoms. For example, glycosaminoglycan and glycoprotein, etc. In animal tissues, extracellular space is occupied by several types of heteropolysaccharides, which form a matrix that holds individual cells together and provides protection, shape, and support to cells, tissues, and organs. Rigid layer of bacterial cell envelope. The peptidoglycan is composed in part of a heteropolysaccharide, which is built from two alternating monosaccharide units. Here you can see detailed structure of different polysaccharides. Say, for example, in case of amylose, there are alpha-1,4 glycosidic linkage between glucose units. This is long chain having alpha-1,4 glycosidic linkage between different glucose units only. This is amylose. The only difference between amylose and amylopectin is amylopectin also contains long chain of glucose unit having alpha-1,4 linkages, but in addition, it also contains branching points with different chain resembling amylose, which is attached to different chain that is at branching point. This is having alpha-1,6 glycosidic linkage. This is structure of amylopectin. Whereas in case of cellulose, there is also glycosidic linkage between different glucose units only, but there is beta-1,4 glycosidic linkage in case of cellulose. That is all about different structure of polysaccharides. That's all. Thank you. Thank you very much.
Thank you, ma'am.
Thank you, ma'am.
Yes, ma'am.
Thank you, ma'am.
Thank you, ma'am.
Thank you, ma'am.